Hi, I'm Morgan Freeman. In the event that an organism may need more DNA, whether it is growing, healing, or just being birthed, the process of DNA synthesis replication is a necessary one. DNA synthesis is the natural and/or synthetic form of creating new DNA strands. First, the enzyme topoisomerase untangles the DNA strand that is commonly referred to as the parent strand. This is done in order to make the process of separating hydrogen bonds between the nitrogenous bases more convenient. After the parent strand has untangled, the enzyme helicase separates the strand into two with the dividing line lying between the nucleotides. Because single strands are not stable enough, single-stranded binding proteins, SSB for short, come in to stabilize them. The newly stabilized single strands of DNA act as the RNA primer for the new DNA strands that will soon be developed. Then, the enzyme primase gathers a strand of nucleotides complementary to those of the single strands. The nucleotide form hydrogen bonds. However, two problems arise at this point. The first is that some nucleotides have not bonded. This is easily fixed by the enzyme known as polymerase. It retrieves nucleotides for the leftover unbonded bases. The second is since DNA replication can only take place from the direction of 5' to 3', we are left with what we would call a leading strand and a lagging strand. DNA is constructed in such a way that one chain is 5' to 3' and the other is 3' to 5'. This is so for the sake of strands being able to match, as many of us know that G goes with C and A goes with T. The lagging strand, the strand that goes from 3' to 5', is required to complete replication in small fragments between each nitrogenous base. These fragments are known as Okazaki fragments. Ligase then connects the Okazaki fragments to the newly synthesized DNA strand. The DNA primer is then eradicated from the cell using a form of polymerase. This form of DNA synthesis/replication is known as semi-conservative replication. Thank you.